To help forests adapt to climate change, scientists are studying which tree species can handle changing environments the best. But how do you go about doing this? With so many moving parts, how do you replicate and even control a whole environment? We have two scientists working out of Fredericton, New Brunswick, who came up with a very clever plan. They built their own automated mini greenhouses, or as they call them, phytotrons.
My name is Rob Vaughn. I'm a forest research biologist. Much of my research focuses on the effects of climate change on the growth and physiology of tree seedlings.
I'm Mackenzie. I'm a master's student collaborating with UNB.
And this project that we're focusing on now looks at the effect of warming and drought on several tree species that are native to the Canadian forest region.
I'm growing my trees here as a part of the TransX trials. So it's an assisted migration trial of multiple coniferous trees looking at the effect of temperature and drought stress. So we're trying to improve stand establishment in the future when we are seeing these warming temperatures and increase in drought. These 12 phytotrons is what they're called. 2 of them are mimicking a site that's been outplanted across eastern North America. So these are all set to mimic those same temperatures and the daylight times as those sites. But being in the phytotrons, we can control the random factors that are happening more closely than we can in the field. So we're looking at the climate sensitivity of these 5 species, such as their response to drought, so mortality, their height growth, their caliper. And we're comparing these experimental temperatures from their seedlot origin temperatures to see how they fare with these increased temperatures.
This is the temperature controller. So the top value you can see is the actual temperature that this chamber is experiencing currently, and the bottom value is what it's set at for this 2-hour duration. So there's a sensor connected to this that runs inside of this recirculation pipe that's collecting air from the center of the phytotron. So it's a good sample of temperature humidity. And that air runs through this pipe into this mixing tub here where the heater is located. And so when this set value gets a degree below the actual program value that it should be at, this relay will engage this heater to warm up the chamber. And if this value gets a degree above the set value it's supposed to be at, the relay will turn on an air conditioner, or the 2 air conditioners in this case, because this chamber is set to one of the coldest treatments, so it has 2 air conditioners. And so that controller will just turn on the air conditioner, the heater, whatever it has to do to warm up or cool down the phytotron to match the set value that it's supposed to be at for that particular treatment.
So this is one of our phytotrons. This is the coolest temperature, so it's mimicking Simon Kush in Quebec. So we have our 5 different species in each of these tubs. So we have red spruce, black spruce, white pine, white spruce, and balsam fir. And within each of these species, we have 5 different seed lots, so meaning that the seed that was used to grow these seedlings came from a different origin. So in each tray here, we have 3 seedlings that are all from the same origin, and then the next row of 3 is from a different origin, and so on until we get to 5 seed lots. So this tub is the tub that would get watered every 3 days. The one behind it is watered every 5. The green tags are watered every 7 days, and this one is our drought treatment that's watered every 9 days. And you can see these white tags here. We're using these for subsets to track our growth, our bud burst, and our bud set over time. So we can see if there's different phenology changes within the different seed lots. So the water's coming through these hoses right here, and it's filling, and it's draining at the same time, but it's draining fast or draining slower than it's filling. So the water will run for 30 minutes, it'll fill the tub, the water will stop, and it'll drain slower. And in each of these phytotrons, we do have each of the 4 drought treatments so that we can monitor and look at the interaction between drought and temperature.
Overall, we're trying to determine the effects of future climate warming on the species and then seed lots within some of those species to try to see how those species would respond to warming over time.
This project is one of many that looks at keeping our forests healthy and productive for years to come. Click on this video to see the great work by researchers in Petawawa as they explore different methods to help forests adapt to a changing climate. Make sure you do all the YouTube things, like and subscribe, and keep an eye out for our next video.